This Week in Virology, the podcast about viruses, the kind that make you sick.
From Microbe TV, this is TWiV, This Week in Virology, episode 1115, recorded on May 17th, 2024. I'm Alan Dove. You're listening to the podcast all about viruses. And joining me today from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. It is 70 degrees Fahrenheit and rather cloudy here. Good day to be inside doing a podcast and not outside getting rained on.
Yeah, it's actually, it's trying to be nice weather here. It's 75 and pleasant, but mostly cloudy. And so we'll see where that goes. And also joining us from Fort Lee, New Jersey, Dixon Despommier.
And now I can legitimately say hello, everybody.
Hello.
The weather, Brianne, is remarkably similar to what you described.
Shocking.
It is shocking. And the other thing is that it seems a little sticky. It doesn't seem like it's a good 70 degrees. It seems like it's a little bit...
It's a wet heat.
Yeah, exactly right. Exactly right. Good day for podcasting, absolutely.
And of course, I'm in Western Massachusetts, so I am the only non-New Jersey participant today.
That's right.
All right, a couple of announcements before we dig into the meat of the episode. A couple of public service announcements. First of all, the Mitchelson Prizes, Next Generation Grants, are annual awards of $150,000. That's not chump change, which support early career investigators who are working to significantly advance human immunology and vaccine and immunotherapy research for major global diseases. These prizes are awarded by the Mitchelson Medical Research Foundation and the Human Immunome Project. Applications being accepted from April 1st to June 9th, so June 9th is coming up real soon. This episode is going to be dropping, and you've got a couple of weeks to get your application together. And there will be a link in the show notes. Also coming up is ASV. If you'd like to register for ASV, you're too late. However, if you have registered for ASV and you're psyched up to go to the meeting, a few announcements about that. First of all, there is still time to sign up for on-site childcare. Enough people have already signed up for it that they are go for on-site daycare. So additional people can sign up past the original April 19th deadline. That's now been extended. If you're bringing your incubator, if you're bringing a child to ASV, you will be able to sign up for that and have them taken care of at the meeting site.
And I would just like to point out, ASV registration is still ongoing.
Sorry.
Early bird registration has ended.
Early bird is closed.
However,
yes.
Standard registration goes until June 12th.
Right.
And you can, in fact, also register late or on site until June 28th.
Right. You're not excluded. I'm sorry, the early bird ended. So you're no longer getting the best possible deal, but you can still register and go.
But as a parasitologist, I must add that there are still probably a few worms still left for the birds that come in late.
Yes.
Probably true.
Good.
Yes, there are always worms.
Good. Yes, indeed.
All right. Second ASV announcement. There are going to be special workshops. There will be information on this in your program PDF and the mobile app and on the meeting website. An example of one of these is it doesn't specify whether this is a dinner or a lunchtime workshop. Oh, it must be a dinner. It's Tuesday at 5:45 p.m. Unfortunately, I guess I'm going to miss this, but it's Think Like an Actor, Speak Like a Pro, Techniques to Create and Deliver Confident, Engaging Presentations. That's by Eleni Kilakos. So there will be several others like that on subjects ranging from education, careers, diversity, and communications to many others. Take a look at those. And finally, if you attended ASV either of the past 2 years, if you went to Athens or Madison, look for and find your name badge pouch. Brianne is helpfully holding it up on the YouTube video here. It is a clear window with a black cloth background that hangs around your neck and holds your name badge.
Has a little pocket in the front and a pocket in the back and a zipper pocket.
Nifty. Yeah, so obviously that's a useful item that you should have hung on to, and if you did, bring it with you, because then they won't have to give you another unsustainably produced piece of plastic to hold your name badge. You can instead use the one from the previous years. All right.
Yes, I think there's one other thing we should probably say before we get into the papers,
Sure.
which is that if you like what we are doing, please consider supporting the science shows of Microbe TV at microbe.tv/contribute.
Yes. Thank you. I think we will announce that again at the end for those who have stuck around, because if you listen to a whole episode, then you really ought to be on board with this. All right. We have a snippet and a paper. We'll start off with a snippet, which I apologize to those of you who are trying to read these papers. They are both paywalled, but they are good papers, so we're picking them anyway. The snippet is a paper from The Lancet called Immunogenicity and Safety of a Booster Dose of a Self-Amplifying RNA COVID-19 Vaccine, called ARCT-154, versus BNT162b2, that's Comirnaty, the mRNA vaccine, COVID-19 vaccine, double-blind, multi-center, randomized, controlled, phase 3 non-inferiority trial. All right, so to parse that, there's a company that, there are a number of people, a number of companies that have worked on developing self-replicating mRNA vaccines over the past many years. And in fact, I believe Pfizer was working on one for COVID-19, but they ended up going with the mRNA vaccine.
Correct.
That they felt was the stronger initial candidate.
Yes, there were a few self-amplifying mRNA vaccines that were under development. The one that is described here, the first author of this paper, by the way, I don't know if we mentioned it, was Yoda, or it's Oda, Y-Oda, so almost Yoda.
Yes. Well, I was just going to get to authors in a moment, but yeah.
Well, the senior author is Walson. And this group has been previously working on this RNA vaccine, ARCT-154, in Japan.
Yes.
And it has actually been approved in Japan and was previously trialed in a trial in Vietnam.
Right.
Can you, before you start in on the meat of this paper, just describe for the people who don't know, including myself, what a self-amplifying vaccine is.
Was just going to get to that.
Good.
By the way, these folks on this paper are from Meiji Seika Pharma, Kitasato University in Tokyo, Arcturus Therapeutics in San Diego, and Walson Consulting in Seattle. Right. So it's an mRNA vaccine like the Pfizer and Moderna vaccines. But with those, what happens is you get injected with the vaccine, and the mRNA dose that goes in is only encoding the antigen, the spike glycoprotein that's going to, give the immune response. In this case, what you're getting with a self-replicating vaccine is the antigen plus the molecular machinery to replicate this RNA. There's an RNA-directed RNA polymerase.
Oh, wow.
Right?
Yeah. So basically, it's just a longer piece of RNA that encodes both the spike and the polymerase.
So how long does that actually go on?
How long does it keep expressing?
Exactly.
That I didn't see in this paper.
They don't specifically address how long it is expressed, but they talk about the idea that it is hypothesized to express for a longer period of time than the traditional mRNA vaccine.
Hypothesized.
But they do not they don't in this study sort of go into the details of how long it's expressed.
And it's still an intramuscular injection?
It's still an intramuscular injection. It's still in a lipid carrier.
Okay.
All of that's the same. The other thing that becomes really useful about one of these self-amplifying vaccines is that you can use a lower dose in the patient, because the dose that you put in will amplify.
Absolutely.
And since we have seen with some of the mRNA vaccines some side effects at administration, perhaps by delivering a smaller dose of nucleic acid, we wouldn't have the same types of side effects.
Yeah.
Do we have any active self-replicating vaccines on the market?
Well, other than attenuated vaccines that have been around for decades.
Right.
I mean,
that's an infection.
OPV, measles vaccine. Yeah, those are actual infectious virus.
Yeah.
There is not another vaccine like this one that is a completely self-contained. It's not actually a virus. It's just the bare minimum equipment you need to replicate the mRNA and produce the antigen.
Yeah, and so this group has been working on this, and so this vaccine that they are studying here is probably the furthest along in previous trials in Japan and Vietnam before the trial that we're going to talk about here.
Right. Right. So as Brianne mentioned, this is a vaccine that has now been approved. I think it was approved in January in Japan. Mm-hmm. And the companies, Arcturus Therapeutics is the company that that developed this. And then they are, I think, going for European approval next, and presumably looking at worldwide approvals. The other advantage of this vaccine, I gather it can be stored at refrigerator temperatures, lyophilized. I read that somewhere?
That was also what I read. I certainly saw in some of the reviews that it was not something that needed to be stored at all at -80. Right. It was not nearly as sensitive as the original mRNA vaccines.
Yeah, so the Pfizer vaccine, that was a major stumbling block, is you can't even just put it in a regular freezer. You have to put it in basically liquid nitrogen in order to transport it, or a lab grade -80 freezer. Right. So this has obviously already, as we said, it's already been approved. So obviously it's already been through phase 3 clinical trials. So why the heck are they doing a phase 3 clinical trial and publishing it now? This is it's a follow-up study where this is not just about finding out if it leads to antibody production. That was the trial in Vietnam. They proved that it works and it prevents, severe COVID-19. Here, it's an actual head-to-head trial against the Pfizer vaccine to see if it's, they don't want to say if it's better, they just want to say if it's not worse. Yeah. It's a non-inferiority trial.
The other thing that's really important about this is you can imagine that as we get further and further from 2020, we can think about making improved vaccines, and that's awesome. But it's harder and harder to test them because people already have some immunity. And so here they're not just testing, is this vaccine non-inferior? They're checking to see whether it's non-inferior as a booster. So for those of us who have already gotten previous doses of a traditional mRNA vaccine, particularly the Pfizer vaccine, which they're mostly studying here, will this work as they do it as a fourth dose in their patients? And is it better or worse than getting another Pfizer vaccine as your fourth dose?
Yes. Yeah, so that is pretty much the trial. They enrolled from December of 2022 to February of 2023, 828 participants, did their demographics and statistics on this group and randomized people, double blind, all the proper controls in here, to receive a fourth booster dose with either the Pfizer vaccine or the ARC-154. So they basically found it's actually a pretty straightforward paper. They're about equally well tolerated. There weren't any particularly bad. They already knew from earlier trials this was a safe vaccine, but this is a larger group, and obviously you track safety. There was one thing that they don't really talk much about, but they mention it. There was a severe adverse event with abnormal hepatic function in the ARC-154 group. They concluded that it was related to the study vaccine, but they don't detail what was abnormal about the hepatic function or what.
Or how they knew that was related to the study vaccine.
Yeah, and why was it considered severe? Did it resolve? I was kind of curious about.
Right. Yeah, they didn't mention that, but it does seem like they were being quite good at covering all of the potential adverse reactions, because they
Yes.
also listed some of the adverse reactions that they deemed not related to the vaccine. For example, one person had an ankle fracture.
Yes.
Which
was deemed not due to the vaccine.
Due to the vaccine. So
Yes.
they're very complete here, but they don't give us a ton of detail about the one severe hepatic function abnormality.
Right. Or, as Brianne said, they don't really even tell us why they thought that was linked to the vaccine, but maybe that's going to be in another paper. They're looking at the immune responses. They looked 4 weeks after the booster, and they found the immune responses to the 2 vaccines, and this is looking at antibody levels.
I was just going to bring that up since I know where we're going with the next paper.
Yes, so the next paper we're gonna harp on that a bit, but here I was actually a little annoyed a couple of times. They say what was it? For the current COVID vaccines, the immune response wanes in the months immediately following administration. I thought the immune response doesn't wane, the antibodies wane. But anyway,
yeah, there were a couple little phrases like that where I was like, I don't know. But
they looked at antibody levels because that's easy to do. And they found that this stimulates a similar antibody response to the Pfizer vaccine against the Wuhan strain and against the Omicron strain. And they concluded that it was actually superior antibody response against the Omicron strain compared to the Pfizer vaccine. But I'm not...
I was very confused by that, to be honest.
Yeah.
Because
the numbers, to me, actually didn't say that, but what was your take?
So my take was, when they looked at it, they had a specific criteria that they would use first to describe non-inferiority. And then if they got non-inferiority, they would go on to look at another criteria that was superiority.
Right.
And it looks like they only did their analysis of superiority on the Omicron strains, and they didn't look at superiority with the original Wuhan strain. From my read of the numbers, the Wuhan strain numbers also met the criteria for superiority. In fact, they met them more superiorly.
Yes. Yeah, it looked... I was looking for, well, why didn't you look at the Wuhan superiority? It seems like it met your threshold, and maybe it's because that strain isn't circulating currently.
I don't know. In their method section, the way they describe it, it sounds like they only did the superiority check the second time around, and I was very confused by that.
Yeah, okay. So I didn't miss something obvious there.
I don't think so.
Yeah. Yeah, so that's oh yeah, so they did mention I think this is actually just in the like the alternative abstract, the research in context thing. They mentioned preclinical studies in mice on active RNA vaccines suggest they may give you better cellular immunity.
Right.
So these authors are aware of cellular immunity. That's good to know.
Yes, and I'm not entirely sure how much a self-amplifying RNA would give you better cellular immunity, but I think that having the thought of how do we get the most breadth in immune responses may be important. So I'm really glad that they are thinking about that. And I'm glad that we're thinking about still trying to improve on these vaccines.
Yeah. Yeah, and this is exactly the kind of trial that one needs to do, especially with so many vaccines and such large quantities of them now on the market. Early in the pandemic, we didn't have any, and then we didn't have enough, and now we are awash in plenty of COVID-19 vaccine.
Right.
And so for any company that wants to enter this, it's become kind of a tough, proposition, especially with the efficacy of the existing vaccines. And that's the point of this trial is, are we as good as they are?
Exactly. And can we be used as a booster? because it's going to be increasingly difficult for us to find a population of people that does not already have some level of immunity.
It's probably close to impossible now.
Right. At least in adults.
In adults, yes. And in kids, well,
Right.
Yeah.
One question I have about this, I think that from the sort of biotechnology, immunology standpoint, this sounds awesome. I wonder a little bit about sort of the messaging. How do we explain this to people? And do people kind of feel good about this vaccine? As I look at it, I feel great about it. But I'm trying to imagine how I would explain it to people I knew who were not scientists in terms of the self-amplifying, given some of the concerns I've heard about other mRNA vaccines.
Yes, that occurred to me as well. I mean, during the whole conversation about these vaccines before this, people with the mRNA vaccines, since those were the first big release of mRNA vaccines in the first place, everybody was, who was not a virologist, was saying, "Oh, gee, is this safe to have? You're changing our cell DNA? No, it's messenger RNA. It's the
Mm-hmm.
So we had to have that whole educational process, and now it's a messenger RNA that replicates.
Right.
So we now have to explain why that's okay.
Right. And how it replicates and why the polymerase is okay.
Yes.
So
and since it's RNA, RNA replicating is not something that's intuitive to most people, even if they remember their high school biology class where DNA does the replicating. I think this is going to be a pretty hard sell in multiple ways, but I think the combination of easier transportation with refrigerator temperatures and lower dose.
I think the lower dose is key.
I think that's a selling point.
I do too.
Even though, perhaps surprisingly in this trial, the side effects and the one thing I was interested in was side effects like malaise, Pfizer flu, which if you've experienced it, you remember these mRNA vaccines were, they're not like a flu shot. It was rough. And apparently that's a thing with these self-replicating ones as well, which kind of makes sense because they're in the same kind of lipid nanoparticle, which is probably what's doing a lot of that. I'm not sure that in itself is going to help, but just telling people that it's a lower dose maybe.
Right. I think the lower dose would definitely be helpful.
Yeah. All right. So that is our snippet. Ask your doctor about ARCT 154. I'm sure they'll come up with some more pronounceable name than that.
Yes, that will. I'm sure there'll be a much better name.
Yes.
But I'm glad to see that.
And I bet it'll be a better name than Comirnaty.
I know. Comirnaty is not.
Sorry, Pfizer, you really didn't do so well with that. But drug names are hard.
They didn't do well with that one. It was BNT162B2.
Yeah.
At least Moderna was, mRNA 1273.
Yeah.
Made more sense.
Yes. All right. Moving on.
If you
we
like
have
vaccines.
Our paper. Since we keep talking about the meat of the episode, this would be the meat of our paper. This is a paper in Nature Immunology called Baseline Innate and T-Cell Populations Are Correlates of Protection Against Symptomatic Influenza Virus Infection Independent of Serology. And this is by co-first authors Robert Medelman and Aisha Souquet. We have co-second authors Leanne van de Velde and Kasi Vegesana. And we have 3 senior authors, Richard Webby, who was on the show last week, Sue Huang, and Paul Thomas. And it also involves work from the Shivers 2 investigation team, which is not the support network for a superhero. It's actually a
Jewish funeral.
The Southern Hemisphere influenza virus something.
Yes. And they are from Southern Hemisphere influenza virus. We'll talk about what it is.
Influenza vaccine effectiveness and response study.
That's it.
And so while most of the investigators here are from either St. Jude's or the University of Tennessee Health Science Center, we do also have some people from the Institute of Environmental Science and Research in New Zealand at the Wallaceville Science Center, which is where we get the Southern Hemisphere members from Shivers. And the data here are really looking at a population of people in New Zealand.
Yes. And Richard Webby is originally from New Zealand and is now at St. Jude. So I'm sure he's a critical link in this whole process. And Brianne, I'm hoping you will take the lead on walking us through this paper.
I am ready to do that.
I thought you would be apt for this.
She's drooling.
I could start with a little context if you like.
Go for a little context,
right.
And then I'll point out some stuff that I was excited about, even from the title.
I was excited from the title too, so this is a paper that is looking at two of the most complicated phenomena in microbiology. It's immunology and influenza epidemiology. And flu epidemiology, we've talked in many episodes of TWiV about why that's a particularly tough thing to track. Every flu season is unique, and so you can't compare from year to year unless you have the same cohort and yada yada. This Shivers 2 consortium is addressing that because they have a huge cohort of people in, I guess, primarily New Zealand, who are participating in this year after year, and they are following vaccine efficacy over the course of years. And the immunology side of it, what they're going to do in this paper is delve into the kinds of the level of detail on the immunological response that we don't normally get in flu epidemiology. It's not just take a bunch of blood samples and see what their titers are, for antibodies. These folks are going to dig deep and see what the correlates of protection, or CoPs, are for influenza, which, yeah, it's right. After all these years with influenza, we still don't know what the correlates of protection are from flu infection.
But that's not just an influenza problem.
No, that's not just...
I think that one of the things that was really cool about this paper and my thoughts about correlates of protection is that they really did some in-depth immunological studies that aren't things that we could have done for many years. And they did this on a large number of individuals and had outcome data. And so typically when I look at different correlates of protection papers, they tend to look at a smaller number of measures in their patients, because it's really hard to look at a lot of measures and a lot of patients. And so in some cases, when we think about correlates of protection, we pick 3 parts of the immune response, or I'm just picked 3. I made that up, but we pick some small number of immune responses and we measure them. And then we say, oh my gosh, one of them correlates with whether or not you get sick. That must be the right one.
Yes.
But there were lots of other immune responses that weren't measured. Perhaps those were actually the things that were protecting you. Perhaps they were the mechanism of protection. And the one you measured in the first place and lucked out on just correlated. And so here they do a really good job at looking very broadly at immune responses in their participants to really find out which of this large number of immune responses that they have measured is related statistically to being protected from disease.
Yes. Yeah, and they do, because even with a large sample, this many variables means that the statistical analysis is not straightforward. You can't just do a linear regression and say, oh, here's the answer. So there's a lot of statistics in the paper, which I felt they explained pretty well.
They did.
And it's all in the service of looking for what really protects people from severe influenza.
Right. And so just as a big picture thing before we go into all the details, because, oh, do I like the details?
Yes.
Part of the reason why any of this is important is that we are trying to make lots of new vaccines against influenza and many other infectious diseases like what we saw in the previous paper. And we can't necessarily always look at all of our individuals and wait and see how many of them get infected. What we'd like to know is, can we measure immune responses in vaccine recipients and know if that vaccine will protect you against infection? And what is the best immune response to measure? So what they're doing here is basically doing this huge measurement of participants to determine which of the many immune responses that they've measured was statistically related to being protected from infection, with the idea of setting up kind of a standard that others could use when they are testing their other newly made influenza vaccines to see, do those influenza vaccines induce this kind of immune response?
Yes. And the reason we need new influenza vaccines, even more than we need new COVID-19 vaccines, is because the current vaccines are the best way we have of preventing flu, and they, kind of suck. I mean, efficacy ranges from 10% in a bad year to maybe 60% in a good year.
That is exactly what they say in their introduction.
Yeah. And in fact, NIAID is putting a large sum of money into efforts to try to develop better flu vaccines, but you can't just throw vaccine candidates at the wall for the reasons Brianne just outlined and hope that something's going to work. You need to know what you're looking for. What do we want to see these vaccines do? And this paper not only gets us an initial read on what the correlates of protection are, it shows how to do those analyses.
Yeah, absolutely.
Yeah.
So previously, when people had tried to figure out things like this, they would generally look at antibody levels, as we mentioned in the previous paper. They would do some serology, or they would just look and say, are you vaccinated or not? Or they would look at some demographic data. And that was kind of all they were looking at. But that didn't really help them. And it also is particularly complicated for influenza compared to a lot of other infectious diseases, because we all have some level of exposure to influenza. So we all already have some immune responses to influenza. So even if you're not vaccinated, you have some response that might make you more or less susceptible. So in this study, they decided to look at 4 groups of individuals. People who were unvaccinated or vaccinated in specifically the 2018 influenza season. And then amongst those 2 groups, unvaccinated and vaccinated, they followed the patients and looked to see who got infected with influenza and who did not get infected. And so they're able to determine what types of immune responses did you have before all of this? And so what kind of immune response did you have that might have been associated with you being protected? What immune response might have been associated with you being susceptible? And so they looked basically before the influenza season at your response, or just post-vaccine, they looked afterwards. So they basically were able, again, big picture thing, they were able to come up with a model that gave them an answer that was better than just looking at antibody levels.
Yes. And so this, we mentioned the sample size. So they started off with a Shivers 2 group and narrowed it to individuals who are 20 to 69 years old in Wellington, New Zealand, who are participating in Shivers 2. And it's about 22,000 people.
Right.
So that's the big pool that they can draw from. And then they selected through a series of filters to get the population that was best to analyze for this type of study. And it ends up being 206 patients, which is a lot fewer. But they've designed this so that they selected the 206 patients who were the best representation of this, and could provide them with the best follow-up.
Right, exactly. So they've got a big number of individuals amongst these 4 groups. When they looked at demographic data, they didn't really see big differences in the demographics of the groups. So that was good. And the first thing that they decided to do was look at the antibodies, because that's what everybody usually does, is look at the antibodies. And they looked at antibody levels in to multiple different epitopes and using a couple different assays. And they found that the assays tended to correspond. The epitopes sort of corresponded, although they did find that one particular antigen was really important. So they determined that antibodies against neuraminidase, particularly the neuraminidase inhibition level, was very important, was both higher in the vaccinated individuals, quite a bit higher in the vaccinated individuals, and seemed to be something that was associated with, a bit associated with their protection.
So these are neutralizing antibodies against the neuraminidase, which is essential for viral entry, right?
Yeah, I was reading the how they did the assay. It ended up being an assay that I'd never heard of before.
Okay.
So it's not sort of a traditional neutralization assay, but it's called an ELA. It's a modification of an ELISA.
Yeah, I was hoping you knew it, but okay.
Yeah, so basically it's an enzyme-linked lectin assay. Neuraminidase is an enzyme that cuts sugars.
Right.
Again, oversimplifying. I know the aficionados will know much more about that information. Neuraminidase cuts sugars. So they co-aplate with a carbohydrate that could be cut by neuraminidase. They then add virus, and they're basically looking to see if the sugar has been cut.
Right.
And then they will add antibodies from their patients to see if the antibodies can block the sugar cleavage.
Right.
So it was a new assay for me, but in the end, it gives you a quantification of neuraminidase antibody titers. It's sort of a functional type of assay.
Yeah.
And again, what they really found is that vaccinated individuals make more antibodies. Shocking.
Who knew?
And that
even I can comment on that.
In particular, people who got symptomatic disease had lower levels of antibodies pre-infection than people who had asymptomatic disease. So it looked like if you had antibodies, you were less likely to have symptomatic disease.
And this is where a lot of previous studies would end.
True.
Right.
Exactly.
That's right. Exactly right.
Yeah, exactly. So it, kind of was like, okay, yes, this is what we expect to see. But what they wanted to figure out was what would happen if they measured some other parameters and whether those other parameters would be better than measuring these antibodies. And so they went ahead and started to do some measurements of a lot of different types of immune cells using flow cytometry.
A lot of different types of immune cells.
Yes. So they looked at many different subsets of immune cells, the frequency of them, whether or not they were producing different cytokines, whether or not they were producing the different proteins on their cell surface. And they looked to see whether having more or less of those, that particular cell subset was associated with protection from disease. They also did this in the vaccinated individuals and in the unvaccinated individuals, so they could find out what is a good correlate with the vaccinated individuals versus the unvaccinated. And they actually point out in one place that they had 11 cell populations that had increased frequency in uninfected people. So it seems like 11 kinds of cells were associated with protection. and 20 cell populations had increased frequency in the symptomatic cases. So 20 types were associated with not being protected. So obviously we are not going to tell you about all sorts of these different cell populations. But they were, then were able to do some statistical measures like looking at odds ratios here. They were able to see things like having circulating T follicular helper cells that were positive for a cell surface protein called ICOS, were associated with protection from disease, particularly in the vaccinated individuals. They saw things like CD4 cells that produced a lot of cytokines, were very protective, like in vaccinated individuals. And there were other things that were less protective in these individuals. They ended up coming up with 2 different profiles of what types of things were protective in the vaccinated and what were protective in unvaccinated. And what they tended to find was that in the vaccinated individuals, looking at many of these T cell types seemed to be quite predictive. And one of the reasons they point out for that is that that's probably because all of these individuals had such high antibody levels that we can't really distinguish amongst them or subset amongst them with antibodies. But if we do actually start to look at these T cell levels, we can start to find people who have who are more likely to be protected. And in the unvaccinated individuals, they could see that having slightly different proportions of inflammatory cells, cells that might be more involved in inflammation, particularly was something that might be involved with you being susceptible to disease.
Right. So the inflammatory cells were worse outcomes, and the CD4, the T cells associated with the vaccinated profile were better outcomes.
Yes. They were even able to come up with sort of threshold levels for each of these different parameters. They used a biostatistical method called the receiver operator characteristic curve, or an ROC curve, to basically look at the trade-off between sensitivity and specificity for each measure, and to come up with what was the exact number that you should look for. So for example, with the CD4 T cells that were making 2 types of cytokines, they found that whether or not you could determine protective or not protected was based on whether you had more or less than 0.022%
yes
of those types of cells. And they were actually able to sort of use their analysis to come up with specific numbers that one should look for each of these populations of cells.
Which is a tremendous tool for vaccine developers.
Right, exactly. And they even were able to look at things like, how well some of these actually looked, or how well some of these were in terms of protection. And again, while serology was the first was really good at showing them who might be protected if a person was unvaccinated, they saw that some of these other measures of looking at T cell numbers were much better in vaccinated individuals in trying to determine who would be protected and who would not be protected. They did another part where they looked at some people who had atypical influenza infections, which they called cryptic lineages.
Cryptic infections, yes.
Cryptic infections. And basically they found that their responses were slightly different. If you looked at my notes, you would see that next to this figure, I wrote cryptic is weird.
Yes. So these are people who appear to have been exposed or infected and didn't have symptoms.
They didn't have symptoms, but then they were positive based on having antibody levels change.
Yeah.
So they weren't necessarily coming up as positive based on symptoms, but it seemed like other evidence suggested that they were infected.
Yeah. Yeah, and they've got a weird profile.
They have a weird profile. So, okay, yes.
Okay. Yeah, it's not, I don't think, they don't really try to conclude anything from that because it's not a lot of cases, and it's...
yeah, I didn't really see much that sort of one could conclude from that.
Right.
Then they, as Alan sort of pointed out before, they measured a lot of types of cells. So if I zoom in on this is like, what's 1, 2, 3, 4, 5, 6. I don't know. This is probably 30, 40 cell types that they were looking at. And they tried to determine whether those cell types could be grouped together. So they could say, we tend to see this cluster of cells all acting in the same way, or something like that, because that would help them determine whether they should measure a particular type of cell or a cluster of cells.
Yes.
And they were able to kind of determine about 13 clusters that they could find. And they were able to even determine sort of which clusters were more important for protection in each of these different groups. And so they specifically found that in the people who did not have infection, or did not have measurable infection, myeloid clusters 1, 3, and 8, and lymphoid clusters 4 and 8, as well as 9 and 13 were higher. And again, the reason why that's important is less that unless you are like me and you really want to think about whether it's CXCR3 or something like that. It's really because this means that another investigator could take a look at these clusters and use them to design an investigation of T cells to determine whether their influenza vaccine was working well. And so this tells you what markers on T cells one ought to look for.
And these are clusters that are probably... or maybe known to be things that the immune system would tend to induce together, or they're doing similar things, or what's going on here?
Yeah. So, if we look at, say, myeloid cluster 1 that they mention, that's a whole bunch of cells that are pretty good at presenting antigen to T cells.
Right.
To turn on a T cell response. A couple of the subsets that they have in terms of the lymphoid clusters are clusters of T cells that are good at helping B cells make antibodies.
Right.
One thing that is sort of notable about all of the different cell types that they look at here was that they look at a lot of different CD4 and CD8 T cell types. They look at some gamma delta T cell types. They look at NK cells, but they don't ever actually measure any B cells.
Right.
So they're only measuring T cells of various types and NK cells. I think they're making the assumption that the serology measure is enough for measuring B cells.
For measuring the B cells, yeah.
Though they do point out in a couple of places that because they're looking at blood, they won't necessarily see things like memory B cells in the lung, which perhaps could be a really great correlate of protection. But I might not give up my lung really easily for these kinds of studies.
It is that's a hard sample to get, yes. So they point out they're not looking at resident B cells. They're not looking at mucosal immunity, which is probably important. But those are things that for this trial, as well as for other trials of vaccines, you're just not going to want to dig into that if you don't have to.
Right. So the thing that's sort of important when thinking about this idea of clusters is that it's not as if all of these huge numbers of T cells or B cells that aren't directly measured here act in isolation from one another. They tend to act together. So we see certain types of T cells are all acting at the same time, sort of as Alan had said. And that's really important for this kind of a study in thinking about the correlates of immune protection. Because technically, what you really need if you're a vaccine developer is to know the correlate of protection. That means that you need something that statistically is present at high level when someone is protected, even if that's not the thing that is doing the protecting.
Yes.
You need a thing that you can measure that is good and that is seen at high levels during protection. And so the idea has always been, well, we need a correlate of protection, even if that doesn't have isn't part of the mechanism of protection.
Right.
And when you look at some of these clusters, you might say, well, yeah, I understand that some of these clusters are things that could be really importantly involved in protection. These could be really, really key for protection. But measuring all these types of T cells is not the easiest thing to do. This is not something that everybody necessarily can do. This is actually not even straightforward flow cytometry. This is intracellular cytokine staining based flow cytometry. So you are taking your T cells, you are stimulating them with peptides from the virus of interest. Then you're trying to see if the cells start to make different cytokines or make different proteins. And so this is relatively tricky.
This is a late game boss fight of cell sorting. Yeah.
Yeah. This is hard. You can even tell in a couple of their earlier figures, if you look at the amount of variation that they see in some of the cell types, it's pretty big because it's really hard to look at those cell types. And so somebody might look at all of this and be like, this is great. This gives immunologists like Brianne something to geek out about. But is this actually better than doing something really simple that could be done in many more places? If I'm developing a new vaccine, is this really going to give me an improvement in my vaccine development compared to doing something that's way easier?
Is this really the most accurate way to predict how somebody's going to do?
Exactly.
Right. And the key word there is somebody, because what's missing here, I know ferrets are not people, so they don't do those. And they've probably got a different set of subsets of cells that work where ours don't work in that direction, but some other direction.
Well, this is people in New Zealand.
Yes.
I see. Of course, I've been there many times, and they're unique and different than everybody else.
I bet they probably have some relatively similar immune subsets.
Yes, I'm pretty sure the immune system is the same.
They are very friendly people, by the way.
Yeah.
I must say that. But I think that, in the old days of biology, to find out if something did something, you took it away.
Mm-hmm.
Right.
And then you saw what happened afterwards, right?
Right.
Like the gallbladder or Kupffer cells or something simple that, everybody could understand. But this is really tough to convince even I think that the vaccine board would be very flummoxed.
Well, this is
the only word I can think of.
So this is why they did the next set of statistical analyses,
right?
Yeah,
which is that they built these random forest models, and I looked this up, and it actually is pretty darn cool. So it's called a random forest because you computationally build decision trees and then train them and see which decision trees, like if this then that type of trees, work best on your training model, and then you put them to a test model of your data and see what kind of model emerges as the best predictor.
Yeah,
basically.
So they're taking the data on immune responses pre-infection, before the flu season, and trying to figure out which model will help them predict whether or not someone got severe disease. And they really had 4 models to start with. One of them was the base model, and that included did the people get vaccinated and how many antibodies did they make?
Yes.
Which is what people have used in the past to try to say whether or not, to predict whether or not you're protected from disease. So that was their first model. That's what everybody has done, sort of the simple thing in the past.
Yep.
The second model was combining those data with looking at the lymphoid cells in their analysis. So only part of the analysis they'd done. They did one that used the base model plus the myeloid or innate immune cells versus the previous was the adaptive immune cells. Or a fourth one was the base, the lymphoid cells, and the myeloid cells all together. And they looked to see which of those 4 models predicted whether or not people had symptomatic disease the best. Does that make sense, Dixon? You're making a face.
No, I'm waiting for the answer.
Oh, okay. Well,
I'm poised to strike.
They found that the base model was basically 79% accurate. And if they looked at just innate immunity, it was also 79% accurate. But if they looked at the lymphoid model plus base, it became 86% accurate. And if they looked at a combination of everything, it was 84% accurate. So basically what they were saying is that if they added this additional data to their model compared to what people had done in the past, they had an increase in how well they could predict, or their accuracy of predicting, symptomatic disease.
Yeah.
And so this was better at telling them if the vaccine was going to protect people.
Yes. It was better at telling them if the people were going to be protected.
Yeah.
Based on what their blood work looked like and what everything looked like before and after the flu season. So they now have a model that is 86% accurate in predicting from your initial blood work whether or not you're going to have serious flu.
Yeah, so that's pretty good.
Yeah, that's very good.
If I was making a vaccine, I would pay a lot of attention to this model, and I would want to test my vaccine to make sure that it was inducing these cells.
I would want that extra 7% of accuracy, yes.
I would also, as a person who is interested in thinking about vaccines, want to take a look at this and say, okay, there are lots of different types of vaccines out there. A little of this came up in the previous paper. You know, there's the live attenuated and the inactivated and the subunit and the mRNA and all of those things. If one particular type of immune cell or one of these types of immune cells is really, really associated with protection, then I should focus on vaccine types that make that kind of immune cell.
Yes.
I shouldn't try to get something that makes other stuff. I should focus on these immune cell types that my model says are predictive of protection.
Right. So this is useful in 2 ways: for testing vaccines that you may already be trying out, and for determining new designs of vaccines. What do you want to stimulate?
Yeah, exactly. And they continued sort of looking at all of their variables. They found that most of the variables that were really important in determining protection were related to what they call cellular immune responses, but really it's types of T cells.
Yes.
And they found that, in fact, the circulating follicular helper T cells that make the protein ICOS were the most predictive and were the most important in all of their groups. And that was true when they looked at their patients by age or sex or vaccination status or however they broke down the groups, whether or not the vaccine induced those types of T cells was the most important thing in determining whether that vaccine might be associated with protection.
Yeah.
And then they looked at this whole thing and they said, well, this might be too complicated.
Right?
It was a nice acknowledgement to see. Yes.
Yeah, it might be really complicated for someone. So what we're going to try to do is we're going to try to come up with a model to determine what is the sort of smallest number of variables you need to measure to be able to look at this. Instead of measuring 40 something types of cells and all of that, what are the sort of minimum measurements you should do to be able to get this high level of accuracy? And they were able to come up with a model that had 19 covariates. So basically 19 different criteria that they would look at. Some of those things are things that are associated with protection. Some of those things that were associated with symptomatic risk. And if you put all of those 19 variables together, then you could get a very high level of prediction from your model.
Yes.
So I presume that a lot of these people, when they move on to a new career, will all become weathermen. Because, you've got more variables there to determine whether it's going to rain tomorrow than whether or not a vaccine is going to work.
Actually, I'm not sure you have.
That's not so bad.
I'm not sure the weather has more variables than the immune system.
Well, we haven't figured it out yet, Alan.
I haven't figured out the immune system either.
Yeah, I'm not sure that we have the immune system figured out either.
That's what I'm saying. When you get equally frustrated, then, there's nothing worse than an immunological weatherman. Because
but at 86% reliability in the forecast, it's pretty
good. No, listen, I don't mean to belittle this work because there's a lot of work here.
It's a lot of work.
There's a little work. There's a lot of work, and the problem is that they're working so in advance of the next epidemic.
Yeah.
So that they're trying to predict which vaccine they can make that will ward it off.
Yep.
Right? It's not which one will work after it's already started, because that's too late.
Well, I mean, the advantage of working on flu is that we get a pandemic every year.
Yeah, right. We get a pandemic every year, and for some reason, I feel like it's been in the news a bunch recently.
A little bit, yeah.
So it's like people suddenly care about flu.
About flu, yeah. We've been talking about it for 3 episodes. This is our 3rd episode in a row on flu on TWiV, and it's a hot topic.
Yeah, and so I think that.
Pardon me, I'm going to have a glass of milk.
So hopefully, the idea is maybe as we're working on that next flu vaccine, perhaps it's an H5N1 or what have you, flu vaccine.
Right.
We can start to now take those H5N1 or whatever flu vaccines that we are developing and look to see if they induce these signatures.
Yeah.
Many people have been developing many of those types of vaccines. We probably need to choose which ones we might move forward into further rounds of production. And this could be a really good way to make those distinctions and try to decide which vaccines will work well.
Sure.
And I've never seen this level of data addressing this question for any other vaccine or for any other virus. At least that's sort of one that we are frequently thinking a lot about. And so I think that this is just a pretty amazing work of immunology in actually answering a question that we all are kind of like, well, antibodies, they kind of correlate. That's good enough. But now we can actually see, well, is it good enough? And would something else be better?
And the answer is yes.
Yeah.
And
something else is not better. It's good to combine with your antibody data.
Yeah, they do actually say that if they use just the lymphoid information without the antibody data, that the lymphoid data is actually as good as antibodies without looking at antibodies.
Right.
So, I think it's sort of a question of, okay, yeah, it's true that we should be looking at other things. We can't look at all those old studies that have always been like, antibodies are good enough, and just assume antibodies are good enough. If we really go into the details, we can see that other things are in fact better. And maybe we should start to measure those things. Maybe we can look at things other than influenza. Wouldn't it be interesting to know if how do those correlates change with different types of microbes?
I was thinking, yes, this could be applied to everything.
Yeah.
No question. But I hate to raise this as a specter at this point, but if you're in industry, if you're in research, this is great. But if you're in industry, you're trying to save every little dime and nickel you can possibly save.
This actually is a great way to save and not lose good projects. So in industry, they want to fail early. You want to know as soon as possible if what you're working on is not likely to pan out. You can kill that project and focus on the others. And this is a great tool. I mean, to have a more accurate, more predictive measure of potential protection. If you've got 5 different things that your lab has produced and you're CEO of a company and you're looking at, oh, wow, we got these 5 different vaccines. That's great, but which are we going to pursue? Hey, guys, can you do this analysis? And then the analyses come back and you say, well, number 3 looks good. Numbers 1 and 4 just cancel, and this could be a very powerful tool.
Right, because in the past, what you would have had is you might have had, okay, we're going to measure all of these for antibody levels.
Yeah.
And maybe we'll look at some stuff about CD4 T cells and CD8 T cells. And we'll find out that number 1 makes these good CD4 T cells and number 2 makes good CD8 T cells. And then you'll have to decide, well, which one do you go with, number 1 or number 2? And your response is kind of like, I don't know.
Or you'll find they all make great antibody responses and you have no idea which one's going to pan out, if any. Yeah. Yeah, this is a way to see better into the future, which I think is going to go over very well in business. To the extent that people are going to pursue new flu vaccines at all, which hopefully they will.
But people are certainly pursuing a number of different, say, universal flu vaccines.
Yes.
I can just see someone opening the door and ripping over his shirt, and he's got a big AI tattooed on his chest. This is a job for artificial intelligence.
Well, that's kind of the direction with the random forest model. That's what they're doing.
That's what I thought about. That's exactly what I thought about.
Oh, yeah. And immunology, if you look at a lot of different types of immunology papers, there are certain types of big data approaches and unsupervised clustering approaches and things like that I as an immunologist have had to learn how to read out of nowhere. It's really funny to me that apparently we all know there's this plot called a TSNE plot that is a machine learning plot that I hadn't learned about, but many other immunologists had, and I was really fortunate that I didn't pronounce it out loud before I'd heard from other immunologists who were like, oh yeah, we all know about this because it's pronounced Tisney. It rhymes with Disney.
Okay.
I'll scrunch my eyes even more on that one.
And so yeah, I think that a lot of those types of models may be things people do with data like this. What I appreciated about this paper is that it used a level of biostatistics that I could understand.
Yes.
It wasn't a black box of AI and machine learning.
And then we did math, and here's the answer. Yeah, no, this actually made a little bit of sense.
I had to get out my biostatistics book, but it was a level that I could understand.
You had the biostatistics book, yes.
My biostatistics book didn't cover any of this stuff. I had the
Nor did mine, but I was able to look it up and get at least the gist of what was being done.
I had the original Snedeker, who invented the t-test. That's way, way in the past. So, you're looking at an old dog that never learned new tricks. Not true, but I didn't learn those tricks.
You have learned a lot, Dixon.
You have learned a
just
lot.
past osmosis.
You have learned a lot. Well, and I think one of my favorite things about this paper is that if we think back across, the past 4 years of discussing COVID-related things,
Yeah.
how many times have we said, but the T cells?
Yeah, no,
Why
of course,
are you
of course,
only
of
measuring
course,
the
of course,
antibodies?
of
Why are you
course,
measuring the antibodies, but the
of
T
course, of
cells?
course.
And here we have really clear evidence that that T cell measurement is important, and in fact that T cell measurement adds something
Right.
onto the antibody
No,
measurement.
I totally agree. I
And
just thought they were.
crucially, we have a paper that not only shows that that's important, it explains how to do it in the most parsimonious way. Because they went that extra step and said, okay, you don't have to do all this stuff that we just did. You can actually simplify it down to just doing this, and you get the predictive power.
Okay.
Yeah, so they come out to, their 19 covariates in the model, and that was sort of a model that would work.
Yeah.
And this paper has just a tremendous amount of data, both in the figures and in extended figures and supplemental figures.
Yep.
And I was, again, because this is stuff I like, I was geeking out at some of their extended figures where they actually, show you how they did analysis for all of these subsets of cells. So you could set up your flow cytometer to do the exact same analysis in the exact same way. They are extremely complete in writing this in a way that somebody else could use this model to look at their flu vaccine and see if that flu vaccine was going to be protective. And I really appreciate that.
Yeah. And they, not just in presenting their methods, but also the code is available. So the paper is paywalled, but you can go on GitHub and download their code if you're into that. And do this analysis on an anonymized test set of data and then apply it to your own stuff.
And so, all of those universal flu vaccines or H5N1 flu vaccines that people are currently working on, they could apply this model and get some idea of ability to protect now.
Yeah. And people who want to extend it to other viruses and vaccines could take this as the approach and say, okay, here's the kind of analysis we need to do for this other virus.
So there's one other link in this chain of information that we haven't discussed too much. And that is that we always assume that these epidemics start somewhere in China, somewhere on a pig farm, where migrating waterfowl have just landed, pooped, and then left. And then now the pig is the mixing vessel, and the farmer gets nailed, and then the next thing you know, the entire village gets nailed, and then the whole Southeast Asian population gets nailed. So why aren't we looking at a vaccine for pigs?
It's a good question.
I mean, on a pig, you could do these studies in advance, and you could actually infect and not infect, and then you could do all the studies, and it probably would turn out to be basically the same as people.
I mean, economically,
No, not economically.
big farmers in Southeast Asia getting a vaccine to all of them to vaccinate their entire herds. I think that's a high hurdle.
Yeah, I mean, I can see kind of two issues with all of that, and some of this, I will admit, I'm thinking less about the pigs, and I'm thinking a little bit about some of the issues with cows right now.
Yeah.
One of the issues is this study, and I'm looking at the figure that I have in front of me. I'm not sure we could do in every single organism. As you mentioned rightly earlier, Dixon, the markers on different cell types in ferrets or whatever other organism would be a bit different.
Exactly.
And there are certain model organisms where we've explained and defined the immune system much better than others.
I agree.
I don't know I have never tried to buy antibodies against cow CD8 or cow TNF alpha.
You've never had a need for it yet.
I have not ever had a need for it.
That
And
is-
so one thing is, I don't know if we could do this level of resolution of an immunology study in some of those other organisms right now. And that leads, of course, to the question of, well, we should figure out how to do that, because Dixon's question is a good question.
Yes.
But right now, I don't think we could do that. I think the other problem is, in some of these places, trying to convince some of the farmers to vaccinate all of their animals might be difficult, in terms of why are you implying that my animals are sick? I don't want to let you come onto my farm and test them, because then you might get rid of all of my herd,
my
and
livelihood.
I'm going to lose my money. And wait, do different countries let me sell my beef if my cow has been vaccinated or not vaccinated? I know there's actually been a little bit of discussion of some of that in beef right now,
which is a huge issue, because countries often use that as an excuse to ban imports, even though there's no evidence that it's a problem.
Yeah, and I think that there are some questions about whether that's going to become an issue going forward. And so I 100% agree with you, Dixon. But my question is just, do we know how to do that study right now in pigs?
Sure.
And could we convince all of the farmers to let us vaccinate their pigs?
Well, I can't think of a country that would be more likely to do that than China, because China can control everything. Basically.
They could potentially do this from central management.
They can.
I think at this point, if we lived in a better world, Dixon, I think your approach would be excellent. Unfortunately, in the world we live in, I think vaccinating the humans is probably the best we can hope for.
Sure. I was just looking, though, at cost benefit, and
yeah,
the cost up front to vaccinate pigs and to figure out the vaccine that works in pigs is X dollars. But what is the overall cost per year, considering 140,000 people in the United States die from flu every year? How much is a life worth? And a lot of other people die in other places too. And then the treatment and hospitalization and all these other costs, when you add it all up, it would probably far exceed any budget that you could imagine for developing this vaccine.
All true, and there probably would be some cost savings agriculturally too.
Yes.
Yeah,
that's actually helping the pigs.
It is.
Yes.
It's almost a one-time only deal. And once you have the model set up, you don't need that many pigs. You don't need that many antigens. You don't need that many. Well, whatever. I don't know.
Yeah.
I don't know. You're right.
I think it's a valid approach, and I would love to see people working on porcine flu vaccines. I think it's a great idea. I don't.
Aren't they vaccinated for some things now?
Gain traction.
I think they are. Although my agricultural vaccine knowledge is certainly limited, but I do think they're vaccinated for some things.
Yeah, I think so. Yeah. Maybe swine flu. The
other thing is, I'm sure. Yeah, I mean, most farm animals are vaccinated for stuff that's common to their species.
Exactly. Yeah. So I spent most of my research life developing a vaccine, believe it or not, for trichinosis, which we then succeeded in doing, and we presented the data to the United States Department of Agriculture, and their response was, well, what are we going to do when we find a positive pig?
Right. Yep, exactly.
I said, excuse me, what do you find? What do you do now when you find a positive pig? I mean, you destroy the pig, right?
Right.
Then their response was, well, then who's going to pay for that?
Right.
And then I just threw up my hands, and so did everybody else, and we moved on to another topic. Basically, it was abhorrent to hear the level of ignorance that they were willing to live with in order to prevent a floodgate of tests for animals like pigs, which carry toxoplasmosis. And then, of course, someone raised that as an issue as well and said, well, you know how many pigs have toxoplasmosis, don't you? And I said, would you mind saying that a little bit louder so that all the consumers can hear you?
Yeah.
Because it's like, in some cases, it's over 50%. So And do you eat any raw pork? No, we don't eat raw pork, and we freeze it. So those two public health measures keep the rate down, at least from them. It's maddening when you get into that kind of an argument. That's all I'm going to say.
No, definitely, thinking about infectious diseases outside of humans, and how we monitor, how we protect.
That's right.
Are all really, really important questions.
You might even say that humans and animals share one health.
Whoa, what?
Somebody could start an organization called that looks at human and animals.
They could start like a field.
Alan, radical, man, radical.
Just a thought.
Yeah.
Just a thought. I hear that.
All right.
So on that front, I know this is a little bit of a side diversion, but it is related to everything that we've been talking about. The CDC this week actually released a new tracking site for influenza in wastewater in the United States. And one of the points that they make about it is that it does not distinguish between human and animal and domestic versus wild animal sources of influenza. But you can see whether, and they also aren't doing H5 versus H3 versus H1, but they are actually able to look at levels of influenza and see if they are kind of particularly high throughout the United States. And there's a dashboard on the CDC's website where you can look at this that was released earlier this week. And it's pretty fascinating to look at.
Sure.
And in fact, there is one dot of really high area in the United States, and it's in Alaska. And as soon as you look at it, you're like, what's going on in Alaska? And what's up? Are they birds? Is it birds? Is it, how are the what flyways are right there? Things like that.
Birds. Could very well be birds.
And so, we're just doing this now in 2024.
Right.
To kind of address some of these exact questions that we're talking about.
And I'm so glad that, one of the things to come out of the pandemic was, wastewater is a good thing to track, to keep track of viruses, which, has come up on TWiV for a few years before that. And good to see.
Yep. I remember the first time I read an article that said, we can tell you to the minute when people in Minneapolis have their coffee break.
Yes.
By
you can measure the
caffeine levels in the Mississippi River,
wow.
just downstream from it. I was knocked out by that.
Oh my gosh, I've never heard that.
You cannot hide anything. Yeah, that's right. So they have 2 coffee breaks, one in the morning, one in the afternoon.
Yep.
You can tell exactly when it occurs. You take the time that you can detect it and subtract 10 minutes because it takes that long for most people to say, excuse me, I'll be right back. So, that kind of data takes advantage of human behavior all the time. And it would be very interesting to see how each of these strains, it's too bad they're not looking at the strains of let's say, COVID-19 to know when the first ones hit. You can detect it almost immediately in the water, and it's just, oh well. Oh well. Yeah. I'm sure the aliens abroad are having a lot of fun with this because, we did that a long time ago. For God, what is wrong with these people? They're people. Oh, that's what's wrong.
Yes.
They just don't want to know, do they?
Yeah.
Yeah, they just don't want to know.
All right. So, I think we pretty well covered this paper.
We did. You guys
perhaps not to the level of resolution that Brianne would like to, but
enough.
If you would like to know about CD107A versus CCR5 versus ICOS, I can tell you, and I could be very excited about it. But my guess was that the listeners were going to be less excited about that.
For the average TWiV listener, I think they've pretty much got this.
Yep.
But it's really cool, and I really hope we see more things like this because this is useful information. Instead of us just talking about stuff, we now actually have some data on it.
Yes, we have correlates of immunity and a procedure for finding correlates of immunity that I hope will be very widely applied. Yeah. All right, shall we move on to some picks of the week?
Yes.
We shall.
Dixon, what do you have for us?
Well, it didn't take me long to find this one, and in fact, I've been sitting on this one for a couple of issues of Science. It's an article whose title is as follows: Nitrogen-fixing organelle in a marine alga. This doesn't sound very exciting, but she's excited.
I think it actually does.
I mean, I'm knocked out by this because of the potential for using this organelle in commercially grown crops. There's no reason in the world why you can't transpose it. I don't even know if it has its own DNA or not. I didn't look at it close. I don't even think the article discussed that. They just discussed its assembly. And this is instead of nitrogen-fixing bacteria, this is an organelle like a chloroplast or a mitochondrion. This is an organelle, an algae that has evolved a way of taking advantage of atmospheric nitrogen to convert it to organic sources of nitrogen. And that is one of the biggest sticking points with regards to supporting outdoor agriculture, because the amount of fertilizer that's used to grow crops nowadays is atrocious.
And that fertilizer is currently produced in a CO2 intensive, non-sustainable manner.
Exactly right. It's a Haber-Bosch type of synthesis.
Which the Haber-Bosch synthesis saved humanity from starving to death during the Green Revolution, but the consequence of that is that we can't just keep doing that.
No, we can't, but we could, but we shouldn't.
Yes.
And the other thing is that, the other thing that Haber and Bosch both reacted to, I think one more than the other, was that they realized that it could be turned into an explosive, because they really tried to do it for agriculture, right? And you'll never guess what the first organic chemical was that was synthesized. Of all the chemicals that have been synthesized, the first one was urea.
Mm-hmm.
Oh.
Again, for the same reason, to create a fertilizer, a nitrogen-based organic fertilizer. So here is the solution, maybe, to our problem of having to deal with runoff and blooms of algae, other kinds of algae, not these.
Yeah, because there's no shortage of nitrogen. The air is like, what, 80%?
It is, but it's
76%.
But it's nitrogen gas.
That's right.
It's dinitrogen gas.
N2.
So the reason why I am very excited, the reason why I was like, is because I teach microbiology.
All right, and so absolutely.
I teach these sort of exact issues in microbiology
absolutely.
Everyone, you're in contact with tons of nitrogen right now,
right?
As you're breathing, but you can't use any of it.
No.
The only nitrogen that can be used, as I told my students in this semester of microbiology, was microbiology that was made by the nitrogen fixing bacteria.
There you go.
Nitrogen that was fixed into a bioavailable form of nitrogen by nitrogen fixing bacteria.
Right.
And so,
was produced by the Haber-Bosch process.
That's right. That's right.
And so this is basically telling, my students I lied, because now there's another way.
Exactly. And it's a microbial way too. It's a microbial
way. But the other thing that's just like so cool about this is we often talk about the endosymbiotic theory.
Yes.
The idea that mitochondria and chloroplasts were once separate microbes.
Yep.
That then were engulfed into some pre-eukaryotic cell and became organelles. And now it looks like there is this third organelle. And so they do look actually at the genome of this organelle. It really does look like...
Okay.
This paper goes into all of those details of, looking at this as a new nitrogen-fixing organelle.
Right.
And sort of the endosymbiosis of it. And so it's like, oh my gosh, the third endosymbiosis.
Yes.
And it allows for nitrogen fixation. And can we figure out how to work this? It's cool in so many ways.
I totally agree. I just got
And even if you
away.
can't put this organelle directly into crop plants, you could potentially use this organelle and these algae or some variation on that in a fermenter to ferment and fix nitrogen at scale and make nitrogen fertilizer without the Haber-Bosch process.
Absolutely right. And without getting an explosive at the end.
Well, yeah, I mean, that's just side benefit.
One of the I used to go out to Alberta a lot to fish, and the couple that I stayed with out there ran a bed and breakfast, and he was involved in the coal mining industry of British Columbia, which wasn't far away from where we were staying. And he said, "I can show you where we actually work." And I'd like to go with you one day and actually see what you do. And he was a welder that fixed the digging instruments, right? So very specialized people that work over there. He said, "Did you see that mountain over there?" And I said, "Yeah." He said, "Well, it used to be over here." I said, "Well, it still looks like a mountain." He says, "That's because we use ammonium nitrate as an explosive, so that not all of it explodes, and enough is left to fertilize and to allow for plant growth." And it's an almost industry dream wish that they could cover up where they were exploding the tops of mountains off like crazy, because they said, no, we didn't do anything over there. Come on, look at how green it is.
It's fine. The trees growing there.
Yeah. Absolutely. But you can dig 3 feet underneath there, and it's a jumble of rock. That's all it is. It's quite amazing. So this, I hope, will offer humanity an out for the sins of the past and create a better world.
It needs a better name, though. UCNYA is not a catchy name.
Well, okay. Well, we can work on this one.
Yes. All right. Brienne, what do you have?
So I have an article that I read the other day that I thought was really cool about using different types of kind of geology mapping tools to actually find a branch of the Nile that used to exist that no longer exists.
Wow.
And so basically what they had realized was that a lot of the pyramids in Egypt are in a weird place. They're sort of in a weird strip of land that's kind of isolated at the edge of the desert. And part of the question was like, why did they pick this weird strip of land to put all these pyramids on? And so in this study, the scientists actually used sort of different types of remote sensing to find where there used to be another branch of the Nile. And so they were able to find this entire additional branch of the Nile.
Oh, this is cool. This is very cool.
And it turns out that all of the pyramids are on the border of this new branch of the Nile. Many of them actually have like causeways or like little docks going to where the Nile would have been. And so it makes the image here is just amazing, where they show up where all the pyramids are and where the branch is, and you look at it and you're like, oh yeah, obviously.
That was riverfront property.
Yeah, that's like
exactly right.
riverfront property. Right. You can of course also imagine that starts to tell you a lot about, how you moved things for building these pyramids, because if there's a river there, that makes it a lot easier. And if all of the pyramids have a little causeway that goes right to the river, that also makes things much easier. But this all just came from kind of radar satellite data, and we're just learning more stuff about the Nile and Egypt. And that's one of those things that I felt like you would have expected. But then they actually were able to go get cores and things like that and really show this whole area was a part of the Nile that we never knew about before.
I'll be darned.
I was
this is so
super
cool.
excited.
Yeah, I would be too.
Neat.
If you want a follow up on this, go over to the next fertile valley, the Fertile Crescent, and you can see how the cities had to move because those two river systems kept changing also.
Yes.
Yeah.
So it's hey, that's the only thing you can say. The only constancy in nature is change.
They didn't have a corps of engineers to channelize and block and dam all these rivers.
Yeah.
No, they didn't. The Peruvians did, though.
Yes. A few civilizations managed to do that. All right.
Wow.
Well, my pick is an NPR article, which is one of these web-based articles that as you scroll, the graphics come up and change according to what you're looking at. It's about the USDA gardening zones, which kind of like Dixon's headline, a lot of people are saying, why do we care about this? So this is a zone map that if you go to the garden store and you're doing any kind of shopping for plants, it'll say grows well in zone 6A through whatever.
Yeah, that's right.
And you can look on a map from the USDA and see what zone you're in. Well, that has changed.
You bet.
Just since 2012.
That's
right. They actually have been updating this over the years, primarily because the climate is changing and warming. And so you can go to the USDA and look at the map, which is, an image, and you can zoom in, and that's all nice. But if you go to this site, you can enter. Let me just put in. I did this earlier, but then I reloaded the page. So if I put in my area in Western Massachusetts, it was Zone 6A in 2012, and we are now in Zone 6B because our lowest winter temperatures have gotten warmer, and so there are potentially more plants that we can grow in this area. But the article does a great job of showing the trends and changing the graphics in the background of the map to explain what they're talking about, and then they link all of this in. They've got a whole story about the other factors and, what are your, what's your rainfall and why don't cacti grow in Alaska and they grow in Arizona. There are actually parts of Alaska that from the zone map, it looks like you could grow a cactus, but in fact, it's too wet, so you can't grow a cactus.
Right.
So just a very cool presentation of the data, I thought.
Yep. Yeah, this website is so cool. I'm scrolling through it and seeing all sorts of great things. I will also say the fact that it has a little cactus that walks around the outside of your screen
yes.
While you're working is also kind of fun.
That's good. That's very cute.
Yeah, they did a lot of cute stuff with it.
If you really want to see how agriculture has changed over the last 50 years, let's say, go to Europe and ask a vintner where they can grow certain grape varieties now. And to compare that to even 50 years ago, the champagne grape has moved. It can now be grown in eastern England near Cornwall.
But it's not champagne.
Yeah,
there.
Then it's not champagne.
Well, that's true. But
it's sparkling wine.
It tastes the same.
Yes. How dare you say that?
I don't know the word for sparkling wine from England.
Yeah.
I can tell you, about Prosecco from Italy and Cava from Spain, but I don't know what the England word is.
Exactly. They tried for years to grow it without success, though. And then all of a sudden they could grow it.
Now they can.
Now they can grow it. And
there are going to be things like that changing all around the world. I mean, at some point we may have trouble growing apples in parts of New England, which is going to really set off a lot of people, I'm sure.
I bet it will. Wow.
But food for thought, and you can scroll through that article and see how your own zone has been affected.
Good deal.
All right, so this has been TWiV 1115. You can find TWiV at Apple Podcasts, Spotify, microbetv.twiv, or microbe.tv/TWiV, or any place else you find podcasts. We'd love to get your questions and comments. Send them to twiv@microbe.tv. And consider supporting the science shows of Microbe TV at microbe.tv/contribute. If you haven't tried the other science shows of Microbe TV, you really ought to check those out too. Thank you to Dixon Despommier.
You're welcome. Again, my brain is overwhelmed with new information. It's true. It's absolutely true.
And thank you, Brianne, who is BioProf Barker on Blue Sky.
Thank you. I learned a lot.
And I'm Alan Dove. You can find me on alandove.com or turbidplaque.com. I find this is always a pleasure. I'd like to thank ASV and ASM for their support of TWiV, and Ronald Jenkins for the music, and Jolene for the timestamps. You have been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.
In fact, to be honest, when I was reading the paper, I was like, oh, I've never seen correlative protection as CoPs. We got to use that.